Hey, it's Mr. Evans. Welcome to Room 310 Biology here at KCHS. Today we're going to be discussing enzymes in this screencast. And enzymes we refer to very briefly in the previous chapter as one of our macromolecule proteins that a function that they carry out. Enzymes are responsible for speeding up life's reactions, and they allow life to happen. They help organisms maintain homeostasis so that a reaction that would normally take a very long time to carry out happens many times in a second even. So let's get a little vocabulary out of the way. By definition, an enzyme is a protein that catalyzes a chemical reaction. Now that definition may not be too useful for you if you don't understand what a catalyst is or what it means to catalyze something. So let's hit that one as well. A catalyst is a substance that will speed up the rate of a reaction, but the catalyst itself is not changed by the reaction. It sounds like one of those old logic problems. All proteins that are enzymes are catalysts. All enzymes are catalysts, but not all catalysts are enzymes. There are inorganic catalysts that will speed up a reaction, but they themselves are not proteins, so we don't call them enzymes. We're only going to focus on the organic ones, the protein enzymes in this section for us. A little bit of history here. We have a couple of famous individuals involved in this. Certainly a long time ago people knew that your digestive system did something to the food that you ate, and there were some famous experiments regarding that. But it really came to this fellow right here, Louis Pasteur. And Pasteur was a Frenchman, brilliant scientist. Invented some of the first vaccines, was one of the founders of microbiology, and Pasteur also is famous for the pasteurization of milk, process that helps keep milk safe. Pasteur was working with yeast one day and trying to find out how yeast could convert sugar into wine, and he reasoned there was something being produced by the yeast that caused that reaction to take place. Turns out he was right. This fellow here, whose name is Buchner, Buchner was able to carry out the same fermentation of sugar into alcohol without living yeast cells. So it's really he that kind of came up with these ideas of specific molecules doing this, enzymes, and he gave us some of the naming terminology that went along with them. Now, how does an enzyme speed up a reaction? All chemical reactions require what's called activation energy. So I have my reactants, my starting points, and I want to produce a product from that reaction. Well, there's this energy hill that we have to get over called activation energy. A way to look at it is you've got a large boulder at the top of a hill, and you would like to push the boulder down the hill, maybe just for fun. You have to add some activation energy, your energy pushing it, to get it to go ahead and roll down the hill. If you want to start a car and drive a car, you need some activation energy from the battery to get that gasoline and oxygen to react that'll ultimately propel your car. So we have a little energy hill we need to get over for a reaction to take place. Enzymes work by lowering the activation energy. This is their key concept right here. An enzyme lowers the activation energy. You can see in the diagram, without the enzyme, the activation energy is way up high. The enzyme reduces the amount of energy required. The reaction will end up going faster with an enzyme than without. So a little bit more terminology, and pay close attention here. This is some key stuff, and we'll do it with this diagram. A substrate is the molecule that an enzyme acts upon. So in this situation we have two different substrates. We have this one and this one. Here's our enzyme. It's a globular protein, and there are areas on it like little pockets called active sites. The substrate will merge or form a temporary association with the enzyme, and we call that our enzyme substrate complex. Our reaction speeds up, it occurs faster, and then we have two different products than the reactants from which we started. And we're going to see this in a couple more slides, but I just want you to get the idea. A substrate is the molecule that an enzyme acts upon. Here's a larger diagram, and here's that good old sucrose that we put in our tea or in our coffee, that disaccharide, and we know it's made out of a glucose and a fructose molecule. Well, we've got our enzyme here, and it's got an empty active site right now. So sure enough, we're going to get a little bit of binding of that sucrose into that enzyme substrate complex. And I see a molecule of water over here. We have to add a molecule of water in here in order to force these two monomers apart. We have to break that disaccharide. So our substrate, sucrose, is now converted into two products, glucose and fructose. Notice that the enzyme remains unchanged. It's simply waiting for some more sugar, some more sucrose, to find its way into that active site so it can speed that reaction up, and so it goes, and so it goes. A good analogy for an enzyme. It helps if you can think these chemical terms, these scientific terms, and put them into some everyday thoughts. And a very, very common analogy for enzymes is a lock and a key. Keys are specific for certain locks. I can't take this key and unlock this padlock over here, and vice versa. I cannot take this key and unlock this door over here. Many textbooks, many scientists will call an enzyme and a substrate fit a lock and key hypothesis. And as seen by this little schematic down here, enzymes very specific, only work with certain substrates. These circular molecules will only fit into these active sites. These more rectangular or square ones will only fit into certain active sites and only work in particular enzymes. How does the enzyme actually speed up the reaction? Well, here's a globular protein. It is an enzyme, and you can already pick out the active site right here where a substrate will fit in. Once it's in there, this becomes a little mini environment separate from the rest of the cell. Maybe the pH is a little different in there. Maybe there's some force or molecular stress applied to sort of torque or stress the bonds of a molecule to break them apart. Maybe you're just aligning molecules in the proper formation so they can fit together. But that active site is a little mini environment, so to speak. Now, can you identify an enzyme? There's a little trick to being able to do this. We look at their names. Here's good old table sugar, sucrose, a disaccharide. And we'll write that word out here. Here is the molecular formula for sucrase, its structure written out. Here's our fructose, and over here is our glucose. For me to digest this in my body, I need to break a covalent bond that is right here and separate those into monomers that I can absorb. So sucrose will be broken down to its monomers by the enzyme sucrase, ends in A-S-E. And you will see that time and time again with enzymes that they end in A-S-E. Look for it. One more example here. I see two monomers. I see a galactose and a glucose. They're joined right here by a covalent bond, and this happens to be the wonderful substance lactose. And you might have heard of people who are lactose intolerant. They can't eat cheese, they can't enjoy ice cream, they can't drink milk. It's because those people are missing, and let me go back one second, those people are missing an enzyme which is known as lactase. And lactase actually breaks this bond right here. If you're missing it, you might feel a little crampy, you might feel some bloating, but you're not able to digest milk properly because you're missing that enzyme. One thing I'd really like you to be careful about, most students get the idea enzymes are only involved in breaking things down. Enzymes are responsible for building up reactions. We call those anabolic reactions. So we have a simple reactant, another simple reactant, and we make a more complex molecule over here. An enzyme is responsible for helping that occur. And this reaction will not take place very quickly without an enzyme to properly align these molecules. So here's a more complex picture of that building up. Wow. Let's see if we can count all the enzymes that we see in this diagram down here of DNA being replicated or copied. Take a moment, look through here. I see an ASE, so here's helicase, one enzyme. Here's polymerase, DNA polymerase, two enzymes. Primase, here's a third enzyme. And let's see if we can find one more in here. Ligase. So we've got a number of enzymes involved in simply copying our DNA every time our cells reproduce. You're building up a more complex molecule that requires enzymes. So what would I like you to take away from this screencast? The bottom line. First, enzymes speed up chemical reactions. 2, the enzyme remains unchanged. It simply hangs around looking for more substrate with which to react. 3, enzymes are very specific. Remember that idea of a lock and a key. Now in class, we're going to be talking about the factors that affect how well an enzyme performs and what might destroy it. Some of you might really enjoy learning about genetic diseases that are caused by the lack of an enzyme, and certainly when we hit the genetics section we'll be learning about quite a few of those as well. So until next time on this wonderful journey of study called biology, we'll see you in room 310.